أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Dear viewers and dear students, I am your host, Professor Dr. Mazhar Ayaz at Veterinary Problems and Solutions, Cholistan University of Veterinary and Animal Sciences, Bahawalpur. Today we will discuss about the Plasmodium, the reasons how it is transmitted, and the vectors are the insects that can transmit the Plasmodium to their hosts, including man, human beings, monkeys, birds, and reptiles, and many other animals, and how the malaria show their symptoms in the human beings. So this is all about today's lecture on the Plasmodium or malaria. Dear viewers and students, as you know that malaria or Plasmodium is transmitted through a vector, and that vector is mostly a mosquito, and among mosquito the Anopheles female plays a vector role for the Plasmodium. So we will discuss all about its diagnosis, its epidemiology, its signs and symptoms, its treatment, and prevalence in the whole world. As you know, the Plasmodium is from family phylum Epicomplexa, and family Plasmodidae, and from genus Plasmodium. So this is a member of a very important disease of man, monkey, and birds. In human beings, it has four species. One is the Plasmodium vivax, other is the Plasmodium malariae, third is the Plasmodium falciparum, and Plasmodium ovale, while there is a Plasmodium avium in the birds. Regarding the life cycle, we have made a model of Plasmodium vivax. So we will discuss about the Plasmodium vivax life cycle. The first of all, the sporozoites that enter through the blood stream through mosquito bite. So they are present in the proboscis of the mosquitoes or in the salivary glands of the mosquitoes, and at the bite site they stay less than few minutes, and in the blood they stay less than an hour, and they enter into the liver parenchyma cells, and here they become the schizones, and that has a special name, that is the cryptozoites. Here in the liver they divide through multifissions and form metacryptozoites. The Plasmodium vivax keep on multiplying while there is a single generation of multiplication for Plasmodium falciparum. The metacryptocytes, they break out of the liver cells, then they pass into the blood streams and enter into the erythrocytes in just 8 to 10 hours after infection. These metacryptozoites, they round up and develop large vacuole in the center that is called ring stage. After staining with the Jimda stain, they appear red nucleus at the edge and thin ring of the blue cytoplasm around the vacuoles, and we will discuss it later on. Here they grow and form the schizones and trophozoites. Here it is very interesting to know that nearly 200 types of Plasmodium species are present, and they have been identified in birds, reptiles, and other mammals. So it is very astonishing and very quick facts. Here if we see at the life cycle of the malarial parasites, The vector, that is mosquito, they transmit the trophozoites or cryptozoites, and that cryptozoites enter into the liver, and here they multiply and they burst out and then enter into the RBCs, and in RBCs again the mosquito take the blood, and inside the mosquito they form the okinoids, and then the sporozoites are developed and then again injected into the human beings. Here the Plasmodium need two hosts. One is the human being where it plays a sexual role, and other is the mosquito where its sexual role takes place. So this is a very important life cycle where we can learn about the control or effective control of the malarial parasites through biocontrol system. As I discussed, that trophozoites they undergo to the schizogony to produce merozoites. Then these merozoites break the cell and enter into the RBCs and repeat their cycle again and again, and their length of each cycle that depends upon the species, like 2 days in the Plasmodium vivax and Plasmodium falciparum and 3 days in the Plasmodium malariae. Then these merozoites are released, and they release the hematin, heme, and ferritin, that is protein, that is hematin, and granules, that waste products is produced by the parasites, metabolites, and these waste products are toxic to the human beings or to the host, and they cause reaction or paroxysm in the host, or the host undergo into the fits. The reason of the product of this reaction is chill in the large muscles, and headache and fever are more common characteristics of the malaria. Then gametocytes are formed. After number of asexual generations, merozoites enter other erythrocytes and develop to the male and female gametocytes, that are macrogametes and microgametes. Here in this picture of life cycle of Plasmodium, we can easily understand that how these uh, uh, infection are caused, and how from sporozoites to gametocytes are formed inside the human cells, and how from gametocytes then into sporozoites or oocysts are formed, or their zygote are formed inside the ticks, inside the mosquitoes. So in this way the life cycle is completed by the two hosts. Here we can divide the various stages of Plasmodium, and in this way we can divide them as asexual stage and sexual stage. Asexual stages are sporozoites, and it is a Greek word that is meant from sporos, means seeds, and their mostly infections is in the form of injection by the mosquitoes. Then is the merozoites, and that is a Greek word also meros, that means the pieces, the stage invading the erythrocytes. Then is the trophozoites, Greek word trophos means nourishment, the form is multiplying in the erythrocytes. And this was all about asexual stages of the Plasmodium. Then there is a sexual stages. Here the gametocytes, that is a mostly sexual stage, male and female gametocytes, and all these stages have their own unique shapes and structures and protein components or complements, so that can also help regarding the differentiation also. In this picture here we can differentiate shy zones of three different species of Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale in a blood slide stained through a Giemsa stain, so that can easily differentiated on their morphometric grounds also. In this slide or blood smear, there is the presence of a number of Plasmodium vivax free merozoites which have been liberated from the confines of their occupied erythrocyte. Here you can see easily the free merozoites. In this blood smear, we have observed the presence of two Plasmodium vivax schizones. One is the immature form, that is on the left side, and other one is the mature form, that is on the right side. The merozoites are undergoing development in these two infected erythrocytes, and they are almost ready to be liberated from the mature schizones. Here in this slide, that is mostly made through thick Giemsa stain, that shows two mature Plasmodium vivax schizones and each containing merozoites inside. Here in this slide, you can observe the amoeboid trophozoites of Plasmodium vivax. In this slide, we can observe the various forms of trophozoites of Plasmodium vivax, Plasmodium falciparum, and Plasmodium malariae and Plasmodium ovale. Here they can be seen. The gametocytes of all four species of Plasmodium can be observed here in this single slide, and on the upper side there is Plasmodium falciparum and Plasmodium ovale, and in the lower side there is Plasmodium malariae and Plasmodium vivax. They have different in the shape and also in the protein. As discussed earlier, the trophocytes, they stay in the RBCs and blood until ingested by the mosquito like female anopheles, that plays a vector role. In mosquito stomach or in the intestines, the macrogametes produce X-flagellates stage, and they fertilize with macrogametes and form a zygote, and that zygote is a motile that is called okinite or oocyst. That okinite penetrates into the stomach wall of the mosquito and grows into the oocyst. Within oocyst there are 10,000 or more spindle-shaped sporozoites, and their size is about 5 micrometer. The sporozoites, they break out in the body cavity and migrate to the salivary glands of the mosquito. Then these sporozoites are injected into the new host in human beings, into the birds, or in the mammals, or in the reptiles with the help of proboscis. Once mosquito is infected, it is always infected and remains infected for its whole life. In avian species Plasmodium avium erythrocytes, the schizogonty doesn't take place in the liver. That is a different life cycle. There is a animated video present in the YouTube. You can use this link to observe it, and you can find how the mosquito transmit Plasmodium and where and how Plasmodium multiply, and then mosquito is infected. Regarding the Plasmodium epidemiology or malarial epidemiology, that is distributed throughout the world, but that is especially present in Asian African countries. The malaria is transmitted by female Anopheles mosquitoes, while the male Anopheles mosquitoes do not produce or transmit malaria. They just feed on the juices of the fruits or nectars. The only female mosquito requires blood for the maturation of its eggs. So in the same way that not only causes anemia to the host like human beings, but also requires their blood for the maturation of their eggs, and in the similar way they transmit the Plasmodium parasite to the human beings. The disease, especially of the warmer climate countries, and more than 400,000 deaths are claimed by the Plasmodium throughout the year, regardless of the age. Regarding the incubation of malaria or Plasmodium, it takes about 8 to 12 days within the host, but can exceed up to 10 months in some cases. So, after 8 to 12 days, the disease is reported, or the signs and symptoms can be observed in the patient, but in some cases it may also show its signs and symptoms after 10 months in some cases also. Regarding the pathogenesis or signs and symptoms of the malaria, so malarial parasites are highly characteristic, fits are highly characteristics. It has various stages. In stage 1, that begin with the severe chill, shivering, teeth chattering for 1 to 2 hours, and even the piles of cloths or quilts or blankets cannot subside the shivering. In stage 2, the temperature is above normal up to 103 to 106 Fahrenheit, and it is transitory in curve shape. In large muscle, chill is followed by burning fever, headache, and sweating while the fever subsides. In stage 3, the symptoms gradually subsides and temperature falls after 6 to 10 hours, and the host they fall in the sleep. And that burning or that shivering is due to the breakage of the RBCs. So destruction of RBCs that cause anemia, and that destroyed RBCs are gathered or deposited in the spleen of the patient. So afterward it may cause the splenomegaly, the enlargement of the spleen in malarial patients. So here we can observe the classical signs of the malaria. So there is a involvement of central nervous system through headache, systemic system with the fever, muscular system with the fatigue or pain, and also the back area with the pain. In the skin there is chills and sweatings, and the respiratory system through dry cough, and spleen with enlargement of the spleen. Stomach, there is nausea and vomiting, and similarly there is involvement of RBCs or blood and liver also. So these are systems they get involved. Even sometimes the eyes are also infected with this, and even the patient, they lose their taste, or their taste can be changed from normal to some bitter or tasteless. So after the number of paroxysms or fits, the attack of malaria subside, but there remains splenomegaly and damage to the hepatocytes or liver cells. Then relapse occurs over a year in the uh, Plasmodium vivax and Plasmodium malariae. There is also a cerebral form of malaria and Plasmodium falciparum. That damage is caused by clogging of the capillaries of the brain, and due to the broken and infected erythrocytes, that is called ghost erythrocytes. Complication of Plasmodium falciparum can be the black water, the black color of the urine. That is due to the dissection of erythrocytes up to 60 to 80% 24 hours. and there is also fever, intense jaundice, hemoglobinuria, and the several attacks are fatal, causing death if undiagnosed or untreated. So this is a very complicated disease. So here in this slide you can observe a 3-year-old boy with severe anemia and dark urine. So that urine is the result from massive intravenous hemolysis, and that condition presents with the severe pallor, jaundice, and passage of dark urine due to the hemoglobinuria, and that may be also associated with the acute renal failure. Here you can see the collected urine at day 0, day 2, and then so on day 3. Regarding the diagnosis of Plasmodium or malarial parasites, various methods has been utilized, and from the very beginning the direct microscopic method was utilized, and you can also access for the uh, presence of Plasmodium parasites in thin or thick blood smears by clicking this YouTube channel or link. Then is the slide stain with Romanowski stains or Giemsa stains that can help us regarding the identification or their presence of schizones or merozoites in the RBCs. In the latest diagnostic methods, PCR is one of them for the detection of the Plasmodium, and the quick methods are immunochromatic test ICT, or there can be the latest agglutination test, and there is also rapid diagnostic test present for the diagnosis of malaria. Here in this slide we can observe the various methods used for the diagnosis of malaria parasites, including blood slides, thick and thin smears, ICT, rapid diagnostic test, PCR. These are methods are helpful for the accurate and timely diagnosis of the Plasmodium parasites. Here in this slide we can also observe the appearance of Plasmodium falciparum in thin blood smears where there are ring shapes, and the ring shapes have their parts that is called inside the blue one part or blue one organs are there called cytoplasm. The host cells are or blood cells are there, they are bluish in color. Then there is a chromatin that is red color, there is a vacuole that is a clear inside, and there are pigmentation of the parasites, and inside there is a stippling also. So in this way we can also differentiate, or we can also name the various parts of the Plasmodium inside. In this slide we can observe on upper left corner there is the electron microscopic picture of Plasmodium. Then on the in the middle you can also observe the gametocytes, and the right one is the life cycle of trophozoites. Then they convert into schizones, then that schizones they are released the merozoites, and in this way they form their whole life cycle up to the early trophozoites in 48 hours. The lower one, that is the complete life cycle of Plasmodium inside the human beings and inside the mosquito, that can help us to understand its life cycle. In this slide, we can observe the various stages of Plasmodium, and this is the first Plasmodium vivax in the thin blood smear, where we can observe it's one of the stage. Here we can see the ring formed Plasmodium falciparum, and it is mentioned to here that Plasmodium falciparum is very dangerous Plasmodium that causes various complications in the host. The Plasmodium malariae is less lethal as compared to the Plasmodium falciparum or Plasmodium vivax. The Plasmodium ovale is not present in Pakistan and India and subcontinent, but it can be present in the Philippines and the Africa. But certain cases has been observed of the Plasmodium ovale in the field practices also. In the thin slides or thick slides, you can observe the signet ring stage, that is after the trophozoite stage, and that can be one of the identification point. The hemeton in the RBCs or the waste of Plasmodium in the RBCs, they form the Schuffner dots, and we are lucky enough that there are two species of Plasmodium that don't form the Schuffner dots. The Plasmodium malariae, they form red colored Zeeman dots, and Plasmodium falciparum, they form the green colored mirror dots. So the only two Plasmodium are left, that is Plasmodium ovale and Plasmodium vivax, they form the Schuffner's dot. So by the formation of merozoites, they look like petals or flowers inside the RBCs, that is called rosette stage. So finally the membrane of RBCs and schizones, they burst after this rosette stage, and all the material is get freed in the blood plasma, and at this stage there is a chill formation inside the body. So there is uncontrolled chattering of the teeth and chill inside the body. After the release of merozoites, the RBCs are bursts, so that bursted RBCs are called ghost RBCs. That empty RBCs are taken up by the spleen, and they are destroyed by the special type of phagocytic cells that are called macrophages, and these cells secrete an enzyme that is a lysolecithin, which destroys ghost RBCs. So macrophages, they destroy with the help of lysolecithin, and in this way the spleen gets enlarged by the abundance of the RBCs. So the spleen is called the graveyard of RBCs. So here the dead RBCs, they gather in the spleen, making splenomegaly by the hyperreactive malarial splenomegaly syndrome. So here you can see the ultrasound and the picture of a patient where the spleen is enlarged on the left side of the body or left side of the abdomen near the umbilical cord. So in this way splenomegaly is formed. With the help of this slide, we can differentiate various species of Plasmodium inside the RBCs of the host. So here we can differentiate Plasmodium falciparum, Plasmodium malariae, Plasmodium vivax, and Plasmodium ovale on their various stages that are present inside the RBCs of the host. The asexual stages of the Plasmodium falciparum can be observed through thin blood smears, and its development can be observed in hours. How they develop inside the RBCs in how many hours, and that is from 0 hour to 48 hours inside the RBCs of human beings. First of all, the tiny rings are formed, then these small rings are formed, then they develop into the large rings, and then there is a development of the early trophozoites, mid trophozoite stage, and late trophozoite stage. The trophozoite then develop into the schizones, and immature schizones, then they develop into the mature schizones. So in this way, the Plasmodium falciparum can be observed from 0 hour to 48 hours in the thin and thick blood smears of the host, like human beings. Here we can observe how the infected mosquitoes can infect to the persons, then the cryptozoites, then they develop into the trophozoites, merozoites, schizones, and then into the gametocytes, and that gametocytes are taken up by the mosquito, where they develop to the zygote, and then they are infected to the another persons. In this way, the life cycle of malaria transmission can be completed. Here, when we get a complete blood cells report from a laboratory other than the slide diagnosis, we find then there is a decrease in hemoglobin or anemia is developing, decrease in hematocrit, there is macrocytosis, and there is a decrease in hemoglobin or HB. Then the white blood cells, they can be increased or decreased. Regarding the biochemistry, there is a hypoglycemia, there is a decrease in calcium, elevated creatinine, elevated LDH, and there is hyponatremia or decrease in sodium. There can be the high lactate and low bicarbonate. Regarding the coagulation, there is a thrombocytopenia, and if we find the blood examination, there is a protein in the urine, that is proteinuria, and blood in the urine, that is hemoglobinuria. So these also help or correlate with the slides or other PCR or ICT tests, so they can be helpful. The post-malarial complications can be the coma due to the cerebral malaria, and that can lead to the death also. There is severe malarial anemia that can also lead to the death, hypoglycemia, and metabolic acidosis or lactic acidosis, acute renal failure, and acute renal failure can result into the creatinine and increase in blood urea, acute pulmonary edema, that can lead to the cough or sometimes the failure of the lungs, algid malaria, that is circulatory collapse, shocks, or peripheral cyanosis. Then there is hemoglobinuria and black water fever or urine in the blood, and that in the final stage there can be the death also. Here in this slide we can observe the malaria retinopathy, where retina in a child with cerebral malaria can be observed, and the macula, there is extensive retinal whitening in the patches, maximal around the fova, that is the center of the picture, and several white centered hemorrhages can be observed. The optic disc is at the right side of the picture also. In this slide, you can observe the fundus of the photograph of an adult with severe malaria showing a large white centered hemorrhages, that is big black arrow here you can see, and there are scattered patches of retinal whitenings in the right arrow, and hemorrhages there also in black arrows also in the right side, and multiple hemorrhages and patches of the retinal whitening in the left eye that can be observed, or these damages can be observed through the malaria in the human beings also. Regarding the treatment, with the development of the time and methods, we have developed ourselves from quinine to the artemetrins, and that took about a century long. Here we have also observed that there is a development of resistance in the Plasmodium also regarding the various drugs. So quinine is also suppressive and curative, but cannot prevent relapses. Then quercetin is better than quinine, but prophylactic against falciparum and suppressive against Plasmodium vivax and malaria. So chloroquine sulfate is curative against Plasmodium vivax, Plasmodium malariae, and also prevent relapses. Primaquine, basoquine, or primethamine, they are best supportive suppressive drugs, and artemetrin can cure all the stages and varieties of the Plasmodium that is newly developed in 2015. Here in this slide you can observe a panorama of anti-malarial drugs, but at the end you can also see the malarial vaccine also that has been claimed in May to June 2020 that this can be used, but that is yeah, under trial in Rwanda country. So after the trial it can be observed inside the market. As it is very promising that about more than 400,000 deaths are claimed by the mosquito, and that is also one of a big number that the mosquito or the Plasmodium claims. So in this map of the world you can observe three regions. One is the endemic, that is the brown, and including them there are certain parts of the Africa and also some parts of the Central America, and then there are certain parts of the Pakistan and India and Bangladesh, China, etc. Then there is the endemic countries where it can be present throughout the year, and the gray area is called the malaria free area. So here you can observe the three colors of the world regarding the malaria. The governments and countries and scientists are working very hard to eradicate the malaria from the world, and in this way the world can be observed in three colors. One is the malaria free, that is green. The green area or the green countries on the map are they are malaria free countries. Then the bluish, they are eliminating malaria. The countries they are working hard to eliminate the malaria from their countries or from their boundaries. And the red one, the people are working for the controlling of the malaria. So neither they are malaria free nor they are eliminating, but they are striving hard to control the malaria from the world. Like many other years, the 2015 was one of the years in which the scientists working against the malaria got their Nobel Prize. So Chinese scientist Yo, she observed the anti-malarial activity from artemetrin, and she developed the medicine from that and got 2015 Nobel Prize. Regarding the prevention of malaria, we have to adopt certain measures like elimination of the mosquito by vector control programs and preventive measures when outdoor between dusk and dawn. We must use full body clothes and also use the nets in front of the doors, and we may also use the mosquito repellents as creams or lotions, and also the living quarters should be the air conditioned so that there must be no any entry of the mosquito, and we can use the impregnated mosquito nets. They are also uh, supplied by the WHO. Even then, we can also use the mosquito coils and also mosquito nets and sprays, and similarly we may close our windows or doors during the day and night hours to avoid the entry of the mosquitoes. Other than uh, medicines, there are phytoremedies that can be used against the malaria. Here we can use the decoction of the cinnamons, we can use the grapefruits, we can use the juice of the ginger. Orange juice can be also helpful against the malaria, and holy basil leaves can also be helpful against the malaria. And then the natural diets containing vitamin C can be helpful to prevent the malaria also. Dear viewers and students, regarding the bird malaria, there are about 5 malarial species like Plasmodium avium, Plasmodium gallinaceum, Plasmodium durii, Plasmodium relictum, and Plasmodium circumflexum that can infect to the birds, to the poultry and other sparrows. Regarding the pathogenesis, they cause serious disease with high mortality in domestic chicken also. Temperature fluctuates, there is development of anemia and splenomegaly. The birds become paralyzed, and that can occur due to the blockage of the brain capillaries. Like the diagnosis of many other protozoas of the birds, the Plasmodium can be observed and identified by the help of schizones in the Giemsa stained blood smears or with the help of PCRs. Their treatment can be the kerosene or chloroquine and anti-malarial drugs. Regarding prevention and control, mosquito can be controlled by narrow mesh nets, mosquito repellents, and impregnated nets. Residual spray of the poultry houses with insecticides like DDT, diazinon, sapramethrin, and lindane can be observed also. The malaria can be observed in more than 109 countries of the world, and every year 25th April is observed as World Malaria Day throughout the world in the recognition of how to control the malaria and eliminate and make the world malaria free. I hope you have observed and learned from these slides, and I hope you will get benefit from these few words. Thank you.